Humans' evolution and cognitive advantages over other species has been attributed to increases in overall brain size, among other neurological factors. A much larger cerebral surface area is generally what sets humans apart from other species, such as rodents, who have lower cognitive and intellectual capabilities. Additionally, certain brain regions that have increased the most in surface area over time through evolution of the species are linked with brain regions that progress the fastest in terms of increased surface area during neurodevelopment of humans. Brain areas that have increased the most in surface area over time through evolution are similar areas to those that expand the most in size during neurodevelopment. These areas of expansion are also the most strongly correlated with the human's intellectual ability. Currently, there is limited knowledge regarding genetic factors that influence these relationships, as well as cerebral surface area development. Human brain structure and size also varies drastically from individual to individual, and the reasoning behind this phenomenon is not fully understood. Previous studies have indicated that cerebral size and cortical thickness are highly heritable. Despite this, there is still a lack of research regarding genetic factors influencing cerebral surface area in children and adolescents. This research is important because the major changes in cerebral surface area occur during development and childhood. Furthermore, previous genetic theories are based on processes during early neurodevelopment which can be reduced in strength over time. Cerebral surface area proved to be highly heritable, with over 85% of phenotypic variants attributed to genetic factors. However, the heritability levels measured varied from brain region to brain region throughout the cortical sheet. The regions of the highest heritability were the medial orbital cortex, precuneus, and the inferior precentral and postcentral gyri. Moderate heritability was seen in the lateral and inferior temporal lobes. Genetic effects were also statistically significant in the bilateral and medial occipital lobes, precuneus, anterior cingulate gyri and sulci, perisylvian precentral and postcentral gyri, and superior and middle temporal gyri. Additionally, shared environmental effects were not statistically significant. Total surface area of the brain was included and considered within the analysis, and with this addition, the heritability of area expansion decreased in most brain regions. This did not change the general trend of heritability, however, and statistically significant genetic effects were observed in similar regions from before, including the occipital and inferior temporal regions. Environmental correlations were also insignificant once again when total surface area was included. Cerebral regions with the highest genetic correlation between global surface area and areal expansion were similar to regions that had high levels of evolutionary expansion as well. These areas included the parasagittal frontal lobe, DIPFC, and inferolateral temporal lobes. The concordance between genetic and evolutionary maps was also statistically significant. Additionally, neurodevelopmental genetic spatial concordance was also significant. Generally, the heritability of cortical thickness was much lower than that of total surface area. However, genetic factors still had a statistically significant influence on determining the phenotype of cortical thickness. The correlation of shared genetic influences on measures of surface area and cortical thickness had variability throughout brain regions. Moderate correlations between global surface area and intelligence score were observed, whereas there were weak but positive correlations between areal expansion and intelligence throughout both cerebral hemispheres. In other regions, such as the left frontal and temporal lobes and middle temporal gyrus, there were strong correlations. However, additionally, there was asymmetry in terms of the strength of correlations between hemispheres, with the right hemispheres having weaker correlations and a lack of statistical significance. The strongest genetic effects occurred in the posterior and parasagittal cortex, according to the research in this study. Previous studies resemble our results in terms of areas with the similar levels of heritability, such as the parasagittal cerebrum and the inferior temporal lobe. However, as the age of the subjects used in previous studies increased, the data skewed farther away from that obtained in this study. The estimates for heritability were much higher and less regionally variable in the brain. These results together suggest that surface area heritability increases with age. Regions in the visual cortex seem to have the highest levels of heritability. This could relate to our evolutionary history, as an enlarged visual system is a major difference between primates and other types of mammals. However, these heritability patterns correspond to visual regions that have evolved the least relative to other primates. There were very strong genetic influences that played a role in total surface area. This level of heritability is consistent with previous studies in terms of children and adolescents. Based on our results, there is supporting evidence for a shared genetic influence on both cerebral surface area and cortical thickness. Previously, it was thought that surface area and cortical thickness were independent of one another in terms of genetics. However, this new research suggests the possibility of genetic coupling between these two factors, especially during early stages of development. Previous studies have found a correlation between a larger cortical surface area and a higher intelligence. Our results support these findings, as regions with the strongest phenotypic correlations are similar to those in previous literature. To conclude, the data gathered indicates that genetic factors influence individual differences in terms of cerebral surface area in children. These genetic factors also subsequently influence behavioral processes and the function of an individual's neuroanatomy. These findings provide knowledge of how genetics plays a role in early neurodevelopment, specifically in terms of cerebral surface area. Applications of this knowledge could be used to study developmental issues in the brain and be used to study factors that influence a child's intelligence. Additionally, these findings regarding genetics also correlate to evolutionary factors that played a role in human neurodevelopment as well, and this knowledge could be used to study human evolution and brain structure over time. Despite the novel findings and knowledge gained from the study, there could be several issues that pose questions about the validity of the findings and the experimental design. The genetic influence on surface area differed heavily between different brain regions, and therefore the conclusions made in the study are not an end-all be-all for proving genetic correlation. Additionally, there are other non-genetic factors that could be influencing cerebral surface area and could have impacted results obtained, as well as the causation of the increased size in some brain areas. Finally, the study focuses on children as its subjects, whose brain sizes are largely variable during this time as they are still developing. Despite using children of similar age, they could be at different stages in terms of neurodevelopment with relation to their peers, which could influence cerebral size or behavioral tendencies such as intelligence.